And we are live. Okay, very good. So it's my pleasure to present you our speaker today, Sophie Imhof, and she will present us the lecture From Patient to Genetics, Paralyzing Struggles of Translating ALS. And I think it's a very nice topic for us to begin our global seminars and lecture series this summer. And without further delays, I will pass the word to Sophie. Please, you're free to go.
Thank you. Thank you for the nice introduction, and thank you for inviting me to giving this speech. I think when you asked me, I thought it was a very, very wonderful idea to do these things, because the talk that I will be giving is something that we usually have in scientific conferences. So at conferences that everyone has access. So I think it's a very nice idea to give these speeches to a wide audience, so everyone can have access to that. So yeah, as was already introduced, I will be talking about ALS. I will be talking a lot about the translation of ALS, a lot of about the clinics, but I will guide you through until the molecular part and the genetics part. So I'm going to start. Okay, this doesn't work. I'm going to start a little bit with who am I? So I'm born Austrian. I was growing up in Lower Austria, and I actually went to a pretty much local university, I would say. And I was studying medical and pharmaceutical biotechnology at this point. So this is my bachelor. But already at that point, I had a major in genetics. So I knew I'm going to stay in science and academia. Within this bachelor, I had the chance to go abroad. So it was my first time actually going abroad. And I went to the north of Sweden. I went to Umeå University. And back then I was still researching on viruses, so very different to what I'm doing right now. And actually the results that I got back then, it was a few years already back, they were published actually last year. So after Sweden, I decided to do a master's. And I went to the Netherlands, I went to Amsterdam, to the University of Amsterdam. And there it's my passion for neuroscience really started. I started with molecular neuroscience, but as I already had it in my bachelor's, I had a major in genetics. And within this master study, I got offered an internship there. So I basically stayed for nearly a year in an internship position at the University of Amsterdam. And I was looking already the first time into neurodegenerative diseases like Parkinson's disease, and on a very neurogenetical side of Parkinson's disease. After that, and somehow within my master's, but also after my master's, I moved to the US and I went to Boston, where I was at the Harvard Stem Cell Institute. Harvard Stem Cell Institute. I started with a literature review, which was published last year. And this is also where my first research started on ALS, basically. So after I finished my literature review, they offered me a position for my own project. So I started actually researching on ALS. And at that point, I was researching on P62 and the entire contribution of autophagy in ALS. And I was using iPSC-derived motor neurons. I will talk about this also later, but I was hired there as a researcher. And then I decided to continue with a PhD. And for several reasons, I didn't want to stay in the US for a PhD. So I moved back to Vienna, and I never actually lived in Vienna, but I moved back to Austria. And I got the position here at the Medical University of Vienna. My PI is actually the head of the ALS center here. So I continued basically pretty much my project that I already started in the US. And I got the chance to add a lot of new things. So here I'm working on the familial ALS with C9orf72 mutation. I'll also talk about this later. For people that are like interested in PhDs, and if you want to continue PhDs, so I'm in the PhD program here in Vienna, and it's the neuroscience PhD program. I am especially lucky, I would say, because I'm a shared student. So I have two PIs. I have Dr. Vanya Naj. She helps me with all the basic research. Her team is really, really good. They are doing, or basically are focused on rare neurological disorders, but it's a really nice team. So that's really helping me in the lab. And then I have my main PI, I would say, that's Dr. Hakan Çetin. He also holds a PhD from Oxford University, and he helps me a lot with the entire setup of the project, a lot of clinical aspects from it. So I'm really lucky with having this very scientific side in my as one of my PIs, and the very clinical side of it. Yeah, so I would say we dive right in. I thought for the people that are not in neuroscience and that are maybe in some kind of other science, it would be nice to give a pretty much short intro into what I'm actually looking into it. So neuroscience is the study of the nervous system, and this seems now like a little bit dumbed down now, but the nervous system, it consists of two main parts, and that's our brain and the peripheral nervous system. So we have the central nervous system and the peripheral nervous system. And I think everyone, if you know something about the brain, people always know that there are different brain departments, and all of these departments have different functions. And I also showed this very nicely here. You can see, yeah, you can see my cursor. I showed this here, and I thought this is a very simple drawing, but I still think it is very nice. Although in the last decade, it was shown that it's not that straightforward. So it's not, though of course, it's the frontal lobe and with speech and smell, but within these departments, it's not like that we only have one cell population. So we start to realize that there are a lot of different subpopulations of cells within these departments. So this is also going to be something very nice for the future to look into. And obviously, all these compartments also can have different diseases. So there are like some main diseases of the nervous system, I would say, and you probably know most of them, but there's something like we call structural disorders. And this is something that combines like injuries, for example, and yeah, after car accidents or any kind of accidents, you can have injuries, and this is something we call structural disorder. Then we have something pretty much very common, vascular disorders, stroke. So that means if the blood clots and the brain is not nutritioned well anymore, and then you have something we call a stroke. Then we come to the main part of the talk today. It's the neurodegeneration, and I think these are the diseases that are all over the news. If there's something over the news, it's neurodegeneration, and the biggest one are Alzheimer's and Parkinson's, I would say, and obviously also ALS. So ALS is also part of neurodegenerative diseases. And then we also have, of course, brain inflammations and functional disorders like headache and epilepsy and all the 1,000 forms of epilepsy. But today I want to talk more a little bit about neurodegenerative diseases, because ALS is one part of it. So basically the biggest ones we have, Alzheimer's, Parkinson, and Huntington, they all pretty much affect different regions of the brain, but the basis of neurodegeneration means that the neurons and the cells in these compartments degenerate. So we lose these cells. And with losing these cells, symptoms start to arise. And before I'm going to start more talking about the clinical side of it, I think in this entire talk that I'll be giving, we need to always remember a little bit the prevalence of these diseases that we're looking into, because ALS is a very rare disease. So if you look here, you have a 100,000 people population, and within these 100,000, only 7 people have ALS, whereas Alzheimer's is a way more common disease. But the thing with ALS is that it's not very, so the importance of ALS is not very much defined by its prevalence, but more, I would say, by its severity. So, because if you look at ALS, we see that ALS is predominantly defined by its loss of upper and lower motor neurons. So we have the upper motor neurons in here and the lower motor neurons, and both of these degenerate. And with this degeneration of motor neurons, obviously symptoms arise, we lose muscle. So the key hallmark of ALS is muscle loss. And as every other diseases, there are different types of ALS. So with different forms, we have on one hand the sporadic ALS, which covers 90% of the patients that we have. So sporadic ALS means that the patients, these 90% have no pretty much obvious reason to develop this disease, in the sense of that there's no familial history. So none of the parents have ALS or any related condition, and they just developed ALS, and we don't really know why that is so. And then we have 10%, so the rest of the patients, that we call familial ALS. And these are the patients that actually have a familial history. So their parents had ALS or FTD, for example. And these are also the ones that are, if we can treat something, then these are the ones in future that we are hoping to treat. Familial ALS also is based on genes, obviously. So you inherit specific mutations in genes. And in ALS, I think it's around 30 genes that are associated now with ALS. And I think if you compare it to Alzheimer's, where it's like hundreds of genes, if I'm correct, so we are we're still a little bit far behind on that. But the the best defined ones, I would say, are SOD1, C9orf72, TDP43, and FUS, whereas C9orf72 is the most common gene that leads to familial ALS. And this also brings me now already into the clinical aspects of ALS and the entire story. So the interesting part also is with ALS that if you develop this disease, it's not like that it starts at one muscle, always at the same muscle, but it also, next to having different types of ALS, we have different onsets of ALS. So that means that patients can, for example, report of they lose not the entire speech, but they kind of their speech is impaired a little bit, or they report that they feel it when they swallow, that things change, and this is something we call the bulbar onset. And the bulbar onset is also kind of not known, but like was reported to be the most aggressive, fast progressing form or onset forms of ALS, which also makes sense because if you imagine that there's a muscle loss, a progressing muscle loss, if it already starts in your throat and in your mouth, it will affect the lung very, very soon. And the lung, obviously, it's essential to our life. So if the lung is affected, then this is also the reason why patients normally a shorter life expectancy than, for example, limbar onset. And a limbar onset is when the patients, weirdly, most of the patients report that they feel the first symptoms when they walk upstairs. So when they walk upstairs, they kind of feel like their legs are not functioning as they should be, and they're not walking up as fast as they're supposed to. And also here on the right, so depending on the onsets you have, you show different symptoms, like a different sequence of symptoms, but at the end, usually they fulfill all of these clinical phenotypes. A very typical one, especially for the bulbar onset, is the atrophy of the tongue. So I think you can see very clearly here the difference to a healthy looking tongue. A lot of times it's bend into one direction, and it's also like the skin or the surface of the tongue is not as smooth anymore as it's supposed to be. In the middle, you have the very typical atrophy look of an ALS patient. So this the shoulders that are moving into the front because you don't have the muscle in the back anymore that can carry them. So this is a very typical look, if you want to call it, of ALS patients. And then we go down here, and this is something I find really interesting. If patients show to have a limbar onset, the first muscle that degenerates is this one. And we don't really know why, but this is something we see. If you see an ALS patient, this is something you most definitely will see. So this muscle degenerates very early. So you can imagine, since it is a very heterogeneous disease, and I think I made that already clear a little bit, diagnosis of this disease is extremely hard. So it is different from country to country. So it's not a universal way of diagnosing ALS, but in the US especially, but also here in Austria a little bit, there is something we call a multidisciplinary diagnosis. That means that you have a patient and there are different specialists sitting together and discussing the pathology. So you really have scientists, clinicians, pharma companies that represent the clinical studies, the physical therapists, nurses, psychotherapists, and they all sit together, they all meet the patient, and only if all of these parties, so to say, if all of these parties then agree that this is ALS, then only then the patient will be diagnosed with ALS. And this is something that arises because it goes after the exclusion principle. If there's always, people always try to find another disease and not ALS because it's pretty much the worst diagnosis you can get. So this is why it takes so long. And sometimes this diagnosis can take a very, very long time. So when patients come into the clinic, it sometimes takes weeks, months, up to years until the diagnosis of ALS. And then when this patient has the diagnosis and he comes into the clinics, they usually come into like 2 to 3 months, depending on the state. And it is very hard to basically monitor their loss of progress, to monitor their disease progression, because they are so individual. So there's something we call ALS functional rating scale, and that's actually accessible to everyone. So it's a web page, we can all use it, and it has different questions on it, and clinicians, everyone uses it. Once you have a doctor's appointment, you go through these questions with the patients and they get a score. And then every 2 months, you see the patients, you can compare that score and see how fast they're losing their muscles or they're losing some kind of skills. Yeah, so this is pretty much the only way there is to monitor ALS patients. And I already hinted it before a little bit, but people are always interested, how do ALS patients actually die? Why do they die? And most of the times, number one death is the lung. So once the lung gets involved, it's a pretty bad sign. And that is also why, for example, lung infections are a very, very bad thing to have if you have ALS, because the lung is something you really need to protect as much as you can, if you can in some way. So lung infection is something that they should really, really avoid if they can. And what they actually die of is a lung failure to some extent, but if we are 100% correct, it's actually not a lung failure. What it is that, so when we breathe in oxygen, we breathe out the CO2, but since the muscles here, so the muscles here all degenerate, these patients are not anymore able to breathe out all the CO2. So that means there will always be CO2 left in the lungs. So they actually die of kind of a toxicity of CO2, and they die pretty peacefully actually in sleep. So they just the lung is simply not functioning anymore. And then we come to the biggest point in clinical ALS: medication and treatment. There is no cure to ALS. So ALS has life expectancy between 3 months and 4 to 5 years. It's very individual on patients. There are patients living with it for 10 years. There are patients living with it for a few months. The medication is very, very limited. So there are 2 treatments out, riluzole and Radicava, whereas one is not in Europe. So one is not approved in Europe, and it's the Radicava, I believe, because they couldn't show the long-term effect. So they tried a lot of times already to get it approved here in Europe, but it wasn't. Also for pretty much good reasons. So it was shown to be not that effective. What these 2 medications or treatments do is still very unknown, but the riluzole, basically what we know what it does is that it delays the onset of ventilator dependence of these patients, because since once the lung is involved, these patients can't breathe anymore by themselves, so they get a ventilator. And with this riluzole, it is believed, or it was shown basically, that this dependence on ventilator is delayed. So they have a life, like a prolonged life for 3 months, which, if you have a diagnosis that says it kills you in a few years, is a lot of times for these patients. Radicava instead is something else. It basically, what it does is, once you have the diagnosis of ALS, or once you have the pathology, it just slows the progression. So it basically inhibits the symptoms to arise for a certain amount of time, but it doesn't give you a lot of time either. So basically, all that can be done with ALS patients is a symptom treatment. And so basically, what you treat are the symptoms that arise, for example, dysphagia, which means so these people can't eat anymore, they can't swallow anymore. And since muscle loss, and we know muscles need energy, the most important thing for ALS patients is to keep their weight, so to eat as much as they can, basically. So there's something we call pack probes, and so they can be fed. It's so once they can't eat anymore, this is an option that most of the patients have to take. Then a lot of times they report stiff muscles, which is also clear because they are sitting at some point the entire day, muscles get stiff. For this, we use physical therapy, which works really well. Muscle twitches, which is not a big burden to patients, I think, but still it can be treated. And then something I will talk about in a second also more, depression. So these patients are highly stigmatized and most of the times also heavily depressed. So psychotherapy is something that is always suggested, and also antidepressants, of course. And next to that, a lot of patients also claim that they're tired, which also makes sense, because if you can't get in all the oxygen anymore, and we know oxygen wakes us up, these patients are more tired than usual, and so there are also ways to treat them. Yeah, and this is something now, environmental aspects. I think everyone, when I talk about ALS, first question was, so what about the environmental aspects? How can it be influenced? What can I avoid so I don't get it? And so, because we're in ALS, it's extra complicated. We really don't know much. What we can really say is that the male gender is a risk factor. So more men are affected by ALS than women are. And this could be because of the early testosterone exposure, but it's also unclear. Smoking, but I think that makes sense. Smoking is bad. Smoking is going to kill you in either way. So it's also toxic, of course, and it can lead to ALS. And then there's something interesting, I think. They showed that ex-soldiers from the US military service are more susceptible to develop ALS than others. And I looked closer into it because I thought this was very intriguing. And they even did a study where they looked at the European military service and they couldn't find any association with ALS. So it must be something in the US, in the military there, that makes them susceptible for ALS. And there are some proposed mechanisms behind that, but it's also testosterone exposure, high stress, physical exercise, very unknown things. And then, yeah, of course, lead and pesticides, but I think that makes also sense because, yeah, they're neurotoxic. One second. And now, as I said before, I'm going to talk a little bit about the psychological aspect, because I think especially in ALS, because there's no way we can really treat them. This is something that we can really do. So psychological aspects, that means we can at least be there as a kind of a therapist. And then I think it's really interesting because these patients go through different stages, and it's very individual for patients. So it's not like that every patient goes through the same. It really depends, but we must always consider that ALS is a disease that we can all get. It's something that comes kind of out of nowhere, and often in a age, I would say, like between 50 and 60, where a lot of people, start retirement. So they work their entire life, they start to settle even more maybe and enjoy life, maybe people would say that, and then they get a diagnosis like ALS. So this is something absolutely devastating, and no one can really understand, I think. And also what I saw a lot was that these patients are obviously completely normal people. Last time in the US, I had a plumber, and I explained to a plumber what ALS is. So this is it hits them hard. And so this is why there's this study, and I think I really like this study, how it started. It shows the stages of the patient. So they talk here about this initial crisis, and I think this is something, of course, once you get the diagnosis, a lot of people are in denial. So there are patients that would, through like the first doctor's appointments, always say, no, it's not ALS. So they try to get into a different diagnosis, basically. So in the beginning, I would say a lot of patients are really in denial. And then they get into this transitional phase where denial goes, they start to see it a little bit more. And that's when the depression starts. So this is when these patients really, really get depressive. And after that, there's a really significant stage, I would say. It's the stage of acceptance. And this is something that's also really emotionally draining to see these patients go through. It costs a lot of courage. And it also says it here, and I think that's very, very true. It costs a lot of courage for these patients to accept that they will die within a few years, a few months. And that leads them even more into depression until they get really to their last weeks maybe of life. And then here in the study, they call it preparatory phase, where these people apparently show to have fear and sadness. And this is something I can really also confirm, the sadness. So if people work with patients, there's a clear difference between depression and pure sadness. So these people stop depression basically and prepare themselves for their last days, their last step. And stopping now with the depressive talks, we're going to go more into genetics now, more into molecular aspects. And I think by the clinical aspect, you can already tell it's a very heterogeneous disease. We see a lot of different phenotypes, and it's going to get worse in molecular aspect. It's going to be even more heterogeneous. So I want to go back to that figure real quick so that we remember. I'm going to be talking now about familial ALS. I'm going to be talking through these genes, most of them, and about the disease mechanism we think they have, about some molecular key hallmarks that we see. So yeah, I think that molecular key hallmarks in ALS obviously is the degeneration of the motor neurons, and then what we see in all the neurodegenerative diseases is protein aggregation. So in all of these types here, we see protein aggregation. Depending on the type, you can have different kind of aggregation. So that doesn't, you can have several protein aggregations, but it still depends, but it definitely mixes. A big topic is also oxidative stress. We see it especially in the SOD1 because the gene that's actually its function. So this is a crucial step, and that's something I'm very excited to be talking about, transposable elements. So transposable elements, I'm going to elaborate in more detail later, but what they are basically traces from viruses that infected our species like 60 million years ago, and they left traces, and we call these traces transposable elements. And these transposable elements, they are in our genome, and they're obviously, our body doesn't want to have viral DNA, so our body tries to inhibit them and defend our, we call it genome defense. But in especially two forms of ALS, we see activation of this viral and ancient elements. So this is something very compelling, and also a lot of research nowadays goes into that. But we'll talk about this in a second. Yeah, and then I want to also talk a little bit about the models that we have in ALS, because I think if you're not in ALS and you work on any other pathology, there are well-established models. You have it for every gene, and they work pretty well. They recapitulate the phenotype, and then you come to ALS, and you don't really have that. So we do have all of them, and I'm going to talk more in detail about them. But of course, we have cell models, we have postmortem tissue, we have the mouse and the rat models. There are also some studies on the monkey models, and of course Drosophila models. But all these models have, of course, up and downsides, and especially in ALS, this can be really crucial for your study. So, for example, postmortem tissue. I mean, I'm working with it myself, and it is great because it is the most disease relevant context you can get. I mean, you get the brain from the patient. You look at the pathology, you look at the phenotype. It's great. The problem is we work in degeneration diseases, so the area that you want to look at is degenerated. That's why they died of this disease. So this is one of the downs. A lot of times when you get the brain, the motor cortex is pretty much gone. A collection of the tissue is always a thing, because it says you need to get it under 24 hours after the death, and it's working pretty well now in 2020, but I mean, it is still a big issue to really get the brains as soon as possible. And then we must not forget that this is a rare disease. So it's not like that you find these patients everywhere. It's very hard to really, I mean, luckily, but it's very hard to get the brains of these patients, because they're just distributed everywhere, and they're not accumulated right here at our hospital. And this is also where we get into the legislations. It's in every country a little bit different. I think it's pretty well made here in Austria. So if they die at the general hospital here in Vienna, then they basically are not forced. I mean, they can say no, but basically their brains are automatically donated to research. But there are so many other patients not dying here in other hospitals and other institutions, and then you need to either really confirm that before that, or you have no chance. Then it's not going to be donated. And then a big issue, a biological issue with postmortem tissue is that it's not manipulatable. So you can't use any gene technology on it, because it's simply something you can look at. So it's something you can look at phenotypes and pathology, but you can't change anything on it. That is why also people started to use animal models, for example, mouse models. And people might disagree with me now, but I think in ALS, mouse models is actually the best established model that we have in ALS. It recapitulates the human phenotype pretty well. You can do behavioral and physical tests with it, so you can actually check if you knock something out, does it help the motor function? So does it move better or faster or whatever? It is manipulatable, so you can do a lot of gene technology on it. Still, of course, there is a difference to the human. We have the same problem that the motor cortex degenerates, but it's less of a problem, I would say, than a minor problem maybe, but you need specific trainings. Not everyone is allowed to work with mice. Me, for example, I don't have the license. And then you have the ethics, especially here in Austria. It's from my feeling, it's really, really strict, which is also good. but so you need to have the ethics on your side as well. And then I want to talk a little bit about iPSC. So for the people that don't know it, it's induced pluripotent stem cells. So basically the idea behind that is that you have your patient, you take skin cells. It's also, you can do it with other cells, but the most common is fibroblasts. And then you make a cell culture out of these. And in the cell culture, you can then differentiate them into any kind of neuron or any kind of cell, not only neuron, any kind of cell that you want. And we can then differentiate them into motor neurons. And then having these motor neurons from a patient, we can do drug testing on it and see what helps the survival of these cells. And by doing this, the ideal situation would be to have a drug development and then go back, so to say, to the patients and use this drug to treat them. Obviously, though there was a big hype about iPSCs, everyone wants to work with them. And I think all the good papers that are coming out right now, they have iPSCs in there. Because I mean, you have a high yield, so that you don't have to really care about that, because you can keep differentiating them and you will always have cells pretty much. It recapitulates the human phenotype to some extent, but I was working with them for quite some while. And for me, especially in ALS, it's very difficult. You need to imagine there are like thousands of researchers all over the world working with these iPSCs, and they all use different protocols. So we use 1,000 different protocols, and we still compare these results and try to find some sense in it. I also think that it's not well established. So we had major issues culturing them. It's not easy. It takes a lot of work and effort to really keep them going. Then also you don't have any behavioral or physical tests, but they are very easy to manipulate. So you can do a lot of gene technology in them. And that's obviously something why it is so hyped. And with this, I think I want to go a little bit, even dive deeper now. We're going to dive now into the genetics of ALS. And as I said before, I'm going to be talking about all of these familial ALS genes, or all of them is the ones that I'm going to present these here. And I want to start with the SOD1 gene. The SOD1 gene, basically, it was the first ALS gene that was found, and it's already 1993. And SOD1 is an enzyme that's responsible for destroying the superoxide radicals in the body. So I think you can already imagine if there is a mutation in it, that can lead to major problems in the cell. And that is also why 185 mutations in that gene are associated with diseases, not only ALS, but also other diseases. And especially in ALS, there are, depending on what mutation that you will see different phenotypes. So it can be, there are very aggressive forms of mutations and others that are less aggressive, which is compelling. It's in one gene. So what did we find? What is the disease mechanism? So first of all, there's a decrease in the enzyme activity. They say 50 to 80%. I think it's variable. And next to this decrease in the enzyme activity, it also has a toxic gain of function. And with these mutations that are in it, a lot of times, of course, it also induces conformational and functional changes. And this then also somehow contributes to the protein accumulation that we see. And the interesting part is that we don't see this SOD1 protein accumulation only in SOD1 patients. We also see it in sporadic patients and other patients. So you will see that also with the other genes I'm going to introduce. There must be something independently wrong with these proteins, because they keep accumulating and also in other types without having a mutation. And ALS, SOD1 is also one of the genes that is now in clinical trials, actually. So there's something we call the antisense oligonucleotide treatment. These are either RNA or DNA single-stranded that you can use to bind the mRNA and to bind these transcripts and make them either, as you can see here, alter the protein, or even make no protein synthesis. So where it would usually produce the protein, so the mutated protein, obviously the dysfunctional protein, with these ASOs, they are trying to alter it, to bring it back to the normal state, basically. And this is also already a clinical study. So this is, I think, by ISIS, but it's not Ionis. They're in phase 1 study right now, and they're using these type of ASOs because there are many different ones, and it's an RNase H, and basically what it does, it binds and it cleaves the mRNA. So it diminishes the toxic gain of function that it gets. At this point, they are evaluating the safety, tolerability, and the pharmacokinetics are still ongoing. But I think at this point, it's the most promising that we have in familial ALS, I think. But it will take a while until this is actually usable. Yeah, then we go into TDP43, and anyone who knows something about ALS knows TDP43 probably. This is the gene that we hear, the protein that we hear a lot with ALS. It's really compelling. There are a lot of different mechanisms that were shown. In general, it was found in 2006, so not even that long ago, and it is an RNA binding protein. And that's also one of the biggest issues that we then see in ALS. I will show you. It's an RNA binding protein, and it helps to regulate the RNA transcripts and the stability, transport, pretty much everything. So it's a really key protein, basically. And for this, obviously, it's localized in the nucleus. And what we see in ALS is that it's not anymore in the nucleus, but it moves from the nucleus to the cytoplasm. So TDP43 is then present in the cytoplasm, and in the cytoplasm, it aggregates. And this also, we don't only see in TDP43, but we see many other forms of ALS. And also mutations in the gene itself are also pathogenic. So it's not only, again, the protein that accumulates, but the mutations in the gene are also leading to ALS. So yeah, here again, as I said that already, so the loss of nuclear TDP43. And with this loss of nuclear TDP43, obviously, you can imagine that then the transcripts that are in there that should be regulated by TDP43 are not regulated anymore. So this is the major dysfunction that it has. Knockout models of it are not viable, and whereas the overexpression models actually show disease phenotypes. And what's also a big issue is that TDP43 lives in self-homeostasis. So that means it regulates its own expression, basically. And you can imagine if it's not in the nucleus anymore, it doesn't do that anymore. So it gets produced even more, and it aggregates in the cytoplasm. So as I already hinted before, I want to talk a little bit more about its RNA function, or like RNA binding function. Now we come finally to transposable elements. So transposable elements are something very, very intriguing, I think. They are, as I said before, viruses, especially retroviruses here. They infected us somewhat like 16 million years ago. And throughout the evolution, these retroviruses, they left retrotransposons, we call them, so transposable elements, and they kind of like jump through the evolution until they reach us as humans. And if you now look at our genome, it's pretty interesting that 45% of our genome are already transposable elements. And like 10 years ago or 15 years ago, no one would have, they called it junk DNA because they thought it doesn't do anything. But now the last few years, this was something that researchers started to look into, and they saw that it actually has functions, and it actually is a big part of our genome for a reason. And this is also where we come into ALS. So since it has a function in our body and TDP is impaired, we see problems with transposable elements. So what basically happens in a healthy person is that there are two ways of genome defense. So our body tries to inhibit these transposable elements. And that happens once in the cell nucleus, where the DNA binding proteins, especially Krebs zinc fingers, they're actually called, they inhibit these transposable elements. And not only are these transposable elements inhibited by their binding, but also by having a very tightly packed DNA, so heterochromatin, the genome is basically preventing their translation. So there is a point where they are in very tightly packed DNA areas. The genome defense is not only happening on DNA level, but actually also on RNA level. And this is where TDP43 comes into play. So as I said before, it is a RNA binding protein. So what it does, it binds these transcripts that are translated and inhibits them from being it binds them from the ones that are transcribed and inhibits them from being translated into some dysfunctional proteins. is what TDP-43 does. And then you need to imagine if this, like here in ALS, goes out of the nucleus and goes into the cytoplasm, these RNA, these transcripts of transposable elements, we also call them long-coding transcripts, they are elevated and they're all over the nucleus and doing whatever they shouldn't be doing. We don't really know that yet. And also what we see in C9, I'll show you that later, is that not only TDP is now missing to inhibit these transcripts, but also the heterochromatin that usually inhibits them from being transcribed now goes into euchromatin. And we see that this genome, and the entire genome structure basically changes and loosens up. And so these transposable elements that should be silenced usually are then activated. And this is still something very unknown. People are actually nearly fighting about this, if TDP is a main key player in transposable elements in ALS. So there are several, I think there are like 5 papers, 3 of them say yes, 2 of them say no. This is one of them that says yes. They say that, or they show, this is, so we have a heat map here. We have 2 controls. I think this is siRNA sham. I think it doesn't target anything. You have here the transposable elements, and they knocked down TDP, and they showed by knocking down TDP that all the transposable elements go up. This is one part of the story. So I mean, I'll show you in a second the second part of the story. So this, people believe that TDP is has the main role in inhibiting transposable elements. But now we're going to C9, and C9 ORF72 is the gene, as I said before, that's the most common gene that leads to familial ALS. It's the gene also that I'm working on. And it's pretty interesting because the mutation that it has is not a single base pair mutation like in a lot of other ALS genes, but it is actually an expansion. So in the dark ones are the exons. This entire thing is the gene. And here you see in the first intron, there is a hexanucleotide repeat expansion. That's how we call it. And this expansion, every one of us has. So also healthy people have it, but healthy people, hopefully we have only like 5 to 10 repeats. And in ALS patients or FTD patients, actually, we see sometimes up to 1,000 repeats. So this repeat expansion is actually what's pathogenic in C9 ALS, because what it does is that it's not only just there, unfortunately, it also gets transcribed. So it gets transcribed into RNA. We call it RNA foci. And this RNA, so these transcripts are then translated into something we call dipeptide repeat proteins. So there is a protein actually coming out of a repeat expansion. And I will talk about this in the next slide, but let me first just tell you real quick about C9, what it's actually doing. So it was found in 2011, pretty recently, I would say. It's important for autophagy and membrane trafficking. And as we know, autophagy is already impaired in ALS, so it's also a molecular key hallmark, I would say. It is the most common inherited cause of ALS, and as I said, it's a hexanucleotide repeat expansion. This repeat expansion then leads to 4 main pathologies. I'm going to guide you through these 3. I think this is pretty simple. This is pretty clear. These are RNA foci. They come from the RNA transcripts, and I'm going to guide you through the other 3 a little bit in more detail. I'm going to start with the dipeptide repeat proteins, actually. So basically, as I said before, we have this expansion in there. And this expansion leads firstly to the loss of the function of the entire gene. But next to the loss of the function, it somehow has the gain of function by producing these dipeptide repeat proteins. And it would be too nice if they wouldn't just do nothing. So obviously, they also have toxic functions. And there are quite some already reported, but I would say the biggest ones are that they obviously aggregate in the cytoplasm, like a lot of other proteins and that disease. But interestingly, very recently, people see that they don't only accumulate in the cytoplasm, but they also accumulate in the nucleus, especially nucleoli, where it was they then interact with DNA and RNA. And last year by Sung, a really, really good paper showed that they don't only interact with them, but they are localized to the heterochromatin especially. So what, people believe with that, now we're going to go a little bit into transposable elements, is that these DPRs that we're now here in the nucleus, these DPRs go down to the heterochromatin, and by the binding of the DPRs to the heterochromatin, this entire genome structure loosens up. We see more euchromatin, and this leads then to the activation of these transposable elements that are usually silenced. So, and I think this is now a second part to the transposable element story. A lot of people, some researchers say it's TDP43 that fails to inhibit them, but this has something to do with the DPRs. So there must be some independent role maybe of the transposable elements in that disease, and we don't see the connection yet. So, but people are trying very hard to prove that right now, including me. But we'll see who goes first. And then I'm going to talk about the third big hallmark in C9 ALS, which is P62 inclusion. So anyone that knows something about neurodegeneration knows P62, because this is a protein that's aggregated pretty much everywhere. You see it here. So this is a hippocampus, human hippocampus, and you have the P62 in green here. You have a GA, which is a dipeptide repeat protein, in red, and then they show the overlap. And what they show is that P62 not only aggregates in the cytoplasm, but it also overlaps with different types of DPRs. Here only the GA, but it also overlaps with the others actually. And if you then look a little bit into the function of P62, and I was working on this protein for 2 years. So this protein is the most multifunctional protein that I've seen until yet. It's pretty much involved in everything. Its biggest role, I would say, is in autophagy, which is also a big problem, because since it aggregates, it can't function anymore in autophagy. So not only that it already aggregates, but also it doesn't help anymore to degrade the other proteins. So everything is just accumulating and nothing degrades itself anymore. And P62 as TDP also lives in self-homeostasis. So since it's not degraded anymore with autophagy, it gets more and more and more. So this is already pretty compelling, but obviously it has to get even worse, because not only the protein, which is translated by QSTM1, leads to a phenotype, to a pathology, but also mutations in this gene lead to ALS. So again, there must be an independent role of this disease, and not just the protein itself, because this P62 accumulates in pretty much all of the ALS types that we see. So yeah, I think I pointed out how heterogeneous everything is, and especially also in molecular aspects, how many different phenotypes we see, how many unclear phenotypes that we see. This brings me now to a pretty current topic in a lot of diseases, but I mean, I obviously see it a lot in ALS. It's this clinical heterogeneity that we also see. So if you see here, for example, I plotted C9 ALS. So these patients are all diagnosed with ALS and with a specific form of ALS, C9. You might think that since these patients already have a very specific type of pathology, they have the same symptoms and the same onsets. But if you look here, I have age of onset, physical onset, and some additional symptoms and life duration. And you can see that it's very, very heterogeneous. So there are C9 ALS patients that are 37 that live for 9 years, and there is a 56-year-old man that lives for 3 years, and some have additional symptoms, some don't have them, some have different onsets. So it's very, very heterogeneous, and this then brings me to a little bit of a provocative thought. If you think that these patients, they all have complete different reasons for developing this disease. So the sporadic people, we don't know really why they develop it. Familial ALS, they have different mutations. They have the diagnosis, and they still get treated with 2 medications that are not very effective. So we treat a spectrum of hundreds to thousands of phenotypes with 2 medications. And I think this is where now the really new and recent research steps in. And there is a really good paper, I think. It's very thought-provoking. It was published last year. And what their aim basically was, is ALS a spectrum of disease, or is it actually one homogeneous disease, which we already can kind of exclude? But they were basically starting to show it might not be only one disease phenotype, and more or less like a spectrum, like for example in autism. So the aim of the paper and their study was to develop a phenotype specific drug, so that we don't classify ALS patients anymore according to their clinical phenotypes that they show, and according to their genetic mutations that they have, but to their molecular phenotypes. And the goal of this is to, for example, if you show that, oh, these patients have the orange phenotype C, we're going to give them this kind of drug because it works against this kind of phenotype. And this is also what they kind of showed. I'm a little bit critical about it, but what they were trying to show is that here you have a very nice, I think, graphical abstract. So they had an ALS cohort, and out of this ALS cohort, they took 148 post-mortem cortices. And these cortices, they then did a bulk RNA. And by using a cluster analysis, they could cluster different kind of subtypes out of it. So they have ALS glia. Basically, they a group that clustered with a high regulation of genes that are associated with glia cells. Then TE is meant with transposable elements, actually. So one subtype with high regulated transposable elements, and one group with high oxidative stress. The problem I have a little bit with this, I mean, it is a great approach, absolutely. But the problem I think that many people have that read this paper is, did they actually pick up different ALS subtypes? Because that's what they think. They think they picked up different ALS cohorts. So within one cohort, they found ALS patients that have a upregulated glia, upregulated TE, or oxidative stress. Or did they just pick up different cell populations? Because they had 148 cortices, and they pretty much mixed everything in a bucket and looked at the regulations. So how do we know that they actually not picked up just the cell population? But at the end of the day, I think this is really, really interesting, and our future perspective also, not just to see different ALS subtypes, but also to maybe use it a little, I don't want to say biomarker, but kind of to mark the stages of disease. You know, are there mechanisms involved in early stage ALS or in later stage ALS? So this would be something amazing to know, because we have no idea in what stage these patients are when they're coming to the clinic. And then, yeah, which I actually only read recently due to a journal club. And I think why I want to present it is because it's a completely different site that they are proposing. Because what they're proposing is that this is a neuromuscular junction. So basically, you have the nerve terminal here, you have the muscle here, and in the peripheral nervous system, you have these nerve terminals binding to the muscles and giving them this, the signal to feel pain, to feel any kind of sensation. And in ALS, it was shown that these junctions are impaired and also less, so they are diminished basically. So their theory is that when they overexpress DOX7, which is a protein that's involved in this activation of all of this, so it's a key player in maintaining these junctions also, that they can use this as a therapeutic target. So they push up the DOX7 and want to enhance the junctions. And they do this because they base their entire hypothesis on something we call the dying back theory. And I think this is why I also want to talk about this paper, because I think this is something really interesting. We believe, or like the common ALS researcher believes, that the first symptom, or the first phenotypes basically arise in the motor cortex. So we look at the motor cortex and the motor neurons degenerate. The dying back theory, though, says that it doesn't actually start here, but it starts here. So it actually starts the junctions. There's something wrong with the junction between nerve and muscle that they degenerate, and then it basically progresses up, and also the motor neurons then degenerate. And what they did basically was they used mice. There's a wild type mouse, and this is a SOD1 mouse. So SOD1 is, as I said before, the gene that was found first to be associated with ALS, and this is a very aggressive kind of mutation that they have here. And then they inject into the tail of the mice DOX7 overexpression vector, basically. And then they looked at the nerve terminals. And I thought here, there is a lot to discuss about it. You have in red, you have the acetylcholine receptors, then you have the nerve terminals in green, and here they have the overlap, and they use this to see the nerve terminal area. And so what you need to look at basically here is the wild type. So these are the healthy ones. These are ALS mice without any treatment. So they actually have ALS, and these are the ALS mice that are treated. And you see here a clear difference. So it obviously it is less, and you can see kind of like an atrophy in the signal also. And then they go to the treatment, and what they show is that, I mean, yes, the signal is definitely higher, but I think what in ALS or in any kind of disease, we always need to remember that what we want is this. We want to have a phenotype that looks like the wild type. And I mean, if you ask me, this does not look like the wild type. Obviously, I thought this was background signal, but I don't think it is background signal. I think it is actual signal. It's just, it's mislocalized even sometimes. And that's when they then looked at the nerve terminal areas. So they looked at this and they measured this. And they said, oh, the nerve terminals are improved. To show it also here in a quantitative kind of manner. But yeah, if you look at this, I mean, obviously this is better than here, but what about all the rest of the signals? So it is kind of questionable. And what they did then was, they were interested, okay, the nerve terminals improved. What about the survival of these mice? Do these mice actually live longer or not? And what they could show is that actually the mice live longer, around 30 days, and that's long for a mouse. So the survival went up, yes. But then they looked into the motor neuron number degeneration. This is the key hallmark that we have in ALS that we're trying to tackle every day. And they had a second graph, I'm not showing it here, but they actually still degenerated. So no matter if they survived longer, for some extent, we don't know what happens, who knows, but the motor neurons themselves, they still degenerated. So obviously this is a great approach, but still we need to question ourselves if the motor neuron's still dying, it's still a matter of time, I guess. But why I like the paper was because what they pointed out was that we will never have a drug that just heals ALS and that cures everything. There will be different drugs. So and we need to combine these drugs. And this is also what they say then in the paper, is that this is an approach to, for example, have a drug that overexpresses DOX7 and at the same time use a drug that then helps to, or is like neuroprotective for the motor neurons. And I think this is something that will be our future also. I don't think we can tackle it in any other way, to be honest. And this is also how I'm gonna get now a little bit to the end of the presentation about the biggest struggles that we have in translating ALS. I mean, I already now pointed them out pretty much now in this presentation, but I mean the major problem that we have is the heterogeneity. So we have it on a clinical level, we have it on a molecular level, on a genetical level. So this is something that we need to overcome in the next years, and not quite sure how we will do that yet. But what also contributes to that is the misclassification. So since we only have these very narrow diagnoses like senile ALS and SOD1 ALS and sporadic ALS, it could be that we misclassify the entire patient cohort. So I think we need to rethink that, and also limited fundings, which sounds funny maybe, but that's actually an issue. We need to basically talk to these grant people and tell them that though ALS only affects 7 people out of 100,000, it is worth investing into this. And obviously, for example, Alzheimer's is way more better established. So it is not that easy to get funding for a disease that not that many people are aware of. And at the end of the day, no one wants to really say it too loud, but yes, I think there's still a very small understanding in what the entire pathology is. So this is also something we have to kind of admit. Yeah, there are some possible solutions, but this is something that personal possible solutions. So this is, yeah, it will take a while until we get there. But obviously, like in all the diseases, we are striving for personalized treatments. So what would be great to go from a patient, look at the phenotype, getting his cells, seeing what actually goes wrong, testing a bunch of drugs, seeing, oh, this drug is working. So we get out a drug and we use this drug on this patient. But I think I don't even have to say all the burdens that there are and all the obstacles that we have to overcome to even make that somehow possible. So I think the solution that makes more sense, or like that's more realistic, is to redefine the patient cohorts, to really find a different way to classify these patients, to not to completely throw away our classification system that we have now and look at it from a completely different perspective, to see if we find something in common in these patients, like with RNA-seq. I think that's a great idea. And then classify them after this. So yeah, this is nearly the end of my presentation. I mean, I think I talked a lot about how terrible the research in ALS and how many struggles that we have, but at the end of the day, I'm going to say something positive, and I think the future is actually promising. And especially in ALS, I like to always think about the quote from Hippocrates, which says that wherever the art of medicine is loved, there's also a love for humanity. And I think that especially in ALS, I worked worldwide on this disease, and there are thousands of really the best researchers, scientists, clinicians, and they work at it day and night we try to cure this disease. And I also like to think that these patients, they don't have a chance to take holidays. So I mean, neither, I mean, we need some holidays, or neither are we trying to take holidays from ALS. And I think ALS is also a field that you don't work in because you want to work on something and you want to be successful, like for personal success. It's something that I think is a huge mystery, and I work in it because I don't want to work on anything else, and I wouldn't. It's the field I want to be in, but it's definitely not a field with high success rates. So actually, the other way. I mean, failure rates are the high failure rates are our daily bread. But still, when you know so little about a disease, I think everything you can do is really to pour your heart and your soul into this. And this is what makes the ALS community so special, that I believe, or like what I saw in the last years, is that no one, like not that many people really work for success in that field, but they really work for humanity. And yeah, this is how I want to say thank you to everyone. And I don't even know where to start. I always want to make this slide. I don't even know how I should start to thank, but obviously all my past educations, University of Amsterdam, Harvard University, the General Hospital in Boston, Massachusetts. Here, the entire team of the Department of Neurology that welcomed me really well and really nicely. And then obviously to the host, that I think this is a great organization you guys have. And I think it's very, very nice that you give people a chance to listen to my to my annoyance of ALS. So yeah, please, questions, remarks, comments.
Well, thanks a lot, Sophie.
Everything.
I really enjoyed this lecture, and I guess all of us. It's especially beautiful to listen this from a fan of the field. And I think for the beginning of the conversation, I will just abuse my rights as a moderator, and I will start with my question. So
Please do so.
regarding this C9orf72 type of ALS, in medical genetics, we know about this group of conditions: expansion disorders, trinucleotide expansion diseases like Huntington or Fragile X chromosome. And in these disorders, we observe such a thing as anticipation, meaning that with next generation, if this condition is passed, it may start earlier and more severe. Do we see something similar here with ALS, with this particular form?
I mean, I'm not an expert on this on the expansion itself, but as far as I know, there's not a study that shows anticipation. It's a really, really good question. I honestly do not know that. But what we see, what's also very telling about this expansion, is that it's a mosaic. So actually, there are we presume that there are different kind of lengths, and depending on the subpopulation of cells that you're looking into. So depending if you're in the motor cortex, so the cells seem to the genome, it has like 1,000 repeats, and in the blood it looks different, so you don't see these repeats. And actually this was also shown in family members, so that some of them, he inherited, the dad had it and he inherited it to two kids, and one of them was completely healthy and the other one had like 1,000 of repeats. So I don't know, but I honestly do not think so, because I don't think so. But that's just my guess.
Well, that's very interesting. Thank you. And we have a lot of people who wanted to ask something, and I saw that Julia raised her hand. I will just repeat a technical moment. So if you are watching us on YouTube, feel free to write your questions in the chat. If you are watching us or participating on Zoom, you can either raise your hand and ask when nobody else is talking, or write your questions in the chat. So please, Julia, your question.
I guess we could give chance other people, but yeah, from myself, I thank Sophie for giving this talk to our organization. And by the way, greeting from Boston. Yeah, we're still on connection. Yeah, very interesting. I don't know. There are some people who ask questions within the chat, or I can start with my own question. Probably.
Go for it, because he was the first one, and then I will just read the ones from the chat.
Like I have few questions, but
let's go.
Yeah. So I'm not clinician myself. Also, I'm trying to explore like clinical side. So I was curious at which stage, at which manifestations, basically, what is the trigger of people when they come to doctor to, like, once the diagnosis is, like, once they got first diagnosed, what triggers?
So you mean what triggers the patient to actually come into the clinic?
Yes, like
so this is something that's, I think, very individual. So I'm not a clinician. I want to say this. I'm not a clinician. I worked with a lot of patients because I'm did a lot of clinical work on it. But it is very, very different to patients. So the interesting thing is you really see the character and the personality of these patients. So I would have patients that were really in denial for a long time, and they would come in once they literally they couldn't move, like they couldn't move their legs anymore, and that were like, okay, I might should see a doctor now. So I think they deny it for a long time, but then are the people for especially if it's interesting, if they have medical background, so if they're say like nurses or anything in that direction, they come in pretty early. And I would say especially if they have bulbar onset, because that's something, you know, everyone hears. You hear it's a very significant change of talking that ALS patients have, I think, and they you just notice it yourself. And I think it's it's a very significant change that you can have. So I would say bulbar onset is something that people look for to a doctor very early.
And then a follow-up question: do you think it's possible to implement screening, like among the more broader population for early stage? Would it be like? What's your opinion? Would it be even feasible? Like if people are coming to their home, like the family doctor, and like if someone would develop a protocol for it.
So on a clinical side, I do not know. I know that on a molecular side, even here at the hospital, there's a project going on that looks for biomarkers, and they're looking at neurofilament. So also what we see is a neurofilament accumulation, and I'm not 100% sure about it, but there are different lengths of it, if I'm correct, and they are trying to basically associated the specific kind of neurofilaments with a stage of disease. And this is something that would be great. So it's really, really hard at this point, but this is something that we will definitely go into future. So neurofilaments I know of. I think for protein aggregation and things like that, it's really hard. I think, well, I wouldn't know how you want to tackle that.
We could give chance other people to ask. I might have more questions unless someone would ask in front. So from chat, like there was this person TFL asked. Thanks a lot, Sophie. A great talk. I have a couple of questions. First, do other disorders with unwinding of heterochromatin also have increased T expression, including, I think, there are some metabolic disorders?
I don't know. It's a really good question. I do not know, but I think it's something also transposable element research is something very novel. So though we know it for a long time that we have them, but as I said before, it's only like a few years ago we started to realize, okay, they're actually also pathogenic, they're doing something. So I wouldn't know of any current studies, but I think that this is that the chances are very high. Why wouldn't it be like this? I don't think it's ALS specific, no.
I might have a comment. Like I also do some transposon research in the past. I worked on PV. You might heard about PV pyranase, but I happen to know another disease which might have this mechanism, and this is one type of muscular dystrophy called FSHD, fascioscapulohumeral muscular dystrophy, which is induced by overexpression of DAX4 transcription factor. And it also includes unwinding of chromatin up to something similar to just zygote after fertilization state, and transposons are getting activated. But how much transposons are involved in pathogenesis, it's still also not known.
Right. How is the disease called? Muscular?
Fascious scapulohumeral muscular dystrophy, FSH, or FSHD. I could type in the chat.
Right. No, I got it. Thank you, because that's interesting. I'm gonna look into it, because let's see, maybe there's something common. Because yeah, I think that it's gonna be pathogenic in many other diseases as well.
So it's like just one transcription factor which makes it.
So that's crazy. Yeah, I know it's.
Thanks a lot. So we have a lot of more questions.
Yes.
The second one was, do ALS patients have any immune phenotypes, e.g. recurrent infections? I'm thinking about generating peripheral neuroimmune interactions. What do you think about this?
Yeah, so actually, yes, there is an immune response that we see, especially clear cells activation of them. The thing with that is that also people are a little bit fighting over this. It's not really known if this is a causative reason. So do these patients actually have immune response initially, and then all of the other pathologies are going on? Or is this something, and this is what I honestly also believe, is it's something that comes with this entire neurons die. Of course, they're gonna also really release some toxic factors, and then the entire immune response starts. But this is not known yet. But yes, there is an immune aspect in ALS. Yeah.
Mm-hmm. And for the moment, let's swap back to YouTube. So we have two questions there. The first one, maybe you can advise literature or articles on how the neuron makes the right specific connections. So it's rather a general question, but maybe you have met something.
How a neuron makes, sorry, what?
Makes a right, well, it's written right concrete connections. I think it meant like the proper connections with muscles, for example, in normal conditions, maybe.
I mean, I have a PI postdoc that it's working here on it, so I can maybe send in the papers later. I don't know them by heart, the papers, but there's a lot of literature in it, especially on the pathology myasthenia gravis.
Mm-hmm.
So if you Google that disease or look it up on NCBI, there's a lot of good literature coming up on these neuromuscular junctions, because it's involved in it, and they look at every factor that is in it.
Mm-hmm, mm-hmm. Thank you. And the second one, do the scientists who done the DOC7 study somehow explain the background red signal in the treatment mice group? Yeah, the one which you presented before and told that it's not
right.
like in wild type.
Yeah, wait, I can't go back. Unfortunately, for some reason, but no, they do not. So that's the interesting part. So they do not explain that. I was looking through the entire paper to find that, and that is why I thought it was background signal first, but it's not. So they actually count this as a signal. And there is also another graph I didn't show where they basically quantified the red signal. And they showed that the red signal in the treated compartment obviously is way higher than the one in the wild type. So it does not at all look like the wild type phenotype, but they definitely counted the entire signal. Yeah.
That's a very interesting point. And
yeah, it is.
We have two more in the Zoom chat. So the first one, thank you for your talk, Sophie. My question is, do you investigate any comorbid disorders that are occurring along with ALS in your research?
So also a colleague of mine, she's also in here. I think she's looking into dementia, and there is one FTD, frontotemporal dementia, and that's actually something that's really interesting with C9 ALS, because it affects the frontotemporal lobe. And we see sometimes in ALS that these patients don't only have motor impairments, so movement impairments, but also sometimes they develop some form of dementia. But I see Teresa just opened her video, so please go and answer for me.
Yeah, actually, thank you, Sophie. So it's called frontotemporal dementia, and it's very interesting because here you also have very different subtypes, as you can see in ALS. So, for example, you have subtypes that affect the frontal lobe, so it's more like the behavioral form, or the temporal lobe, so it's more speech disorder. And in those dementia forms, in the behavioral dementia forms, you see very often in kind of 25% of familial cases, you see that C9orf72 repeat expansion. And it's very interesting because before there was actually no overlap between ALS and FTD, because in one disease with the or as we know now, with the same mutation, you have one of them affecting the motor neurons and one affecting the frontal neurons, or in the frontal lobe. So it's not known actually what causes these different phenotypes. So it's thought that there may be some additional mutations. For example, in FTD, it could be that there's an additional mutation in the MAPT gene. And in ALS, there's an additional mutation in the SOT gene, and this is some kind of modifier, but actually it's not known yet. But it's very interesting because there are some mixture of phenotypes, ALS, FTD, and it's 88% have that C9orf mutations, or up to 88%, I think.
Wow, thank you. It's very interesting also. And I think it's also the question for you too. Why does ALS selectively affect motor neurons and non-sensory neurons? Is there any mystery around this? Do we know something?
This is the question I got actually by my PI when I started my PhD. This was his question, like if I get the PhD, it's the question that why are motor neurons so susceptible to get these phenotypes, and we have no idea. People like the common, or like what people answer is always, yeah, they are more vulnerable. So you would say that the motor neurons are more vulnerable and it just hits them the first, and since they die so fast, these patients, it doesn't even get to the other neurons. So this is one of the theories, but we don't know yet what these motor neurons have and the others don't have, that it just really doesn't affect anything else.
Sophie, I have a question. So, like to your answer, actually. So, could it be because of differential expression of some of the possible candidate proteins which are involved in the mechanism, like this P62? Like, what is known about expression of all these proteins, like P62, the SOT, and everything? So could it be?
So well, that's my project. I will look at that, the different regulation, different expression of these proteins in affected and unaffected areas. So to see what's actually the problem. But the thing is that especially P62, as I said, it's a multifunctional protein, so it's expressed everywhere. And yeah, so I don't think it's the dysregulation maybe of P62, but I agree. I think I'm pretty sure it must be something about that. But I think we're talking here about like an epigenetical issue and not only genetic.
Well, there are good news and bad news. Bad news, we haven't provided a direct answer to your question, unfortunately, but the good news that we just defined a big field inside ALS that maybe you can also try to fill.
Exactly, great. Let's not
Yes.
say that.
So right now we have a small pause, and I will use this moment to send the Instagram of Sophie. I think you show this as the last slide, and especially if you have some questions after the lecture or after the stream, which maybe you want to ask directly, you can probably write here or there. And also, I would mention that I was really surprised by the risk factor of serving in the US military, making the service. So do you have any idea what can be behind? I mean, some billing options, or maybe I have no idea, basically.
I don't dare even asking to. No, also paper, they elaborate a little bit on the possible mechanism. And I mean, we do, if we think about it objectively, yes, the military in Europe is probably not as well, the people like soldiers here are maybe not as well trained, if I may say that, than they are in the US. So yes, so they go to a complete different training, and I think that this might be an issue that that they actually go through more mental stress than they would go in Europe. But then again, I don't know about the military in Europe. I know it in Austria, but I think the US is doing something wrong.
Could it be like participation in the missions, like in Asia, in Middle East, and so on?
Yes. They say that even. They say that these are broad missions, so it's extra stress, and they show that it wasn't significant that the ones that were abroad, but it looked like a pattern.
But they might be even exposed to some pesticides, like during Vietnam War, like what happened, and maybe. just
that's true. I didn't even think about it. Yeah, that's true. It could be something like this. Also, who knows what's in all of these bombs and like weapons that they use? I mean,
There's also another question: if it's a causative agent, I mean the pesticides or some risk factors which the U.S. military is facing, or maybe all the stress is just a trigger to the condition which could develop because of some genetic predispositions or something.
it's a very good question, because I don't know about the U.S. military service, but about the smoking, for example. So there's this discussion that obviously smoking could be a causative agent because smoking is bad, but it could also be that actually you develop the disease because you just developed the disease, and because the lung is so important that additionally you have this disease. The smoking also kills your lung even more. And you start off with a complete different threshold already as if a non-smoker. But that's also, I don't know. We don't know.
One more question. So among those potential biomarkers and triggers of ALS, what do you think about participation of tau protein, which is also involved in Alzheimer's and in Parkinson as well? So it's something common, and it could also lead to induction of transposable elements, according to some people.
I just wanted to say there was this paper last year that showed that it entangled it, and then the transposable elements were upregulated. Yes. To some extent it would be great, but I think if we look at tau pathologies, it's more homogeneous than here. So yes, I think it maybe for like a cohort and like a specific type, but Teresa, back me up if you know better, but I think that it's just too heterogeneous in ALS to use it effectively.
Could it be like tau aggregation forms everywhere, like in Alzheimer, Parkinson, ALS, but the region of the brain is different?
Yeah, definitely. It's a very probable option, to be honest.
All right. So if you're still here with us on Zoom, and right now you have a beautiful opportunity to unmute yourself and to ask a question, because otherwise we have a lot of more topics. Okay, I see Antonina unmuted yourself, please.
Yes, can I have some question? In molecular therapy was published one article when American genetics use CRISPR-Cas system, and they say that it is a new way to cure this disease. Do you use it in your research, this CRISPR-Cas system?
I do not use it in ALS. I don't use CRISPR-Cas in ALS. I have a colleague that uses it in a different kind of pathology. For ALS, I mean, the thing is with CRISPR-Cas, but people might disagree. It is just not too well established at this point, I think, to really use it as a drug therapy in ALS. So I think we have way better paths to follow, some like overexpression drugs or something else, than we have with CRISPR-Cas at this point, because CRISPR-Cas, the system itself, is already so complex and not well established yet, I would say. So that we then combine this with a super complex and heterogeneous disease. I think it's gonna be I don't want to say impossible, but at this point maybe.
Okay, thank you.
So I can only add that, of course, I'm very biased because I'm a big fan of gene therapy, but I just think that, CRISPR-Cas itself as a therapeutical system has a lot of problems around, starting from the delivery and up to the immune response, immunization to the system, etc. But
did you watch the show on Netflix about the gene therapy?
No, not yet. Can you recommend it?
You need to. It's a CRISPR-Cas. Unnatural selection, it's called. They talk all about CRISPR-Cas and the biohackers in the US and how they deliver CRISPR-Cas, and that it's just it's a really, really good show. It's actually with the guy from MIT. How is he called? Kevin something, that developed CRISPR-Cas.
I see.
Fen Chan, his name.
Yeah.
From the Broad Institute.
Yeah, exactly.
So here's plus one take-home message for you guys. Just
exactly, watch Netflix.
Yeah, there's a weekend right now, so a good point. You can go and watch it, and I think I will join you.
It's definitely worth it. It's definitely worth it.
I have a follow-up question. So talking about your research, is it okay to ask what's your primary model object? So are you using patient samples or IPS differentiated neurons or animal models? So what's your main object?
So I'm lucky again that I'm working in the hospital. So we have access, also Teresa, we have access to patient samples, which is great. I also have collaborations with Barcelona, so I'm gonna get more brain samples from them. I'm not working with IPCs for a reason. I'm not the biggest fan of them at this point. And I'm developing my own cell line actually at this point with induced DPRs. And we have another collaboration with Sheffield, and I'm gonna get some already established cell lines from them. It's NSC34 cells with inducible expansion. So yeah, these are the models that I'm using. I'm not using mouse. I don't have a license actually for animal models, and I think for my hypothesis and for my study, the post-mortem tissue is perfect. That's really essential.
Animal models overall, they have, as I understand, a lot of problems with modeling the neurodegenerative diseases because they end up being convergent on symptoms and biomarkers, but often they don't respond to the drug. So there was this Alzheimer drug trials failure massive last year. So probably the same with ALS. Might be the same.
Oops, something happened. You are muted for some reason, Sophie.
Do you hear me again?
Oh, yeah. Yes.
Yeah, it said the host muted me.
Oh, we should find this host.
Yeah, I think we have the same problem in ALS because if we have a mouse, like I showed before, with a SOD1 mutation, we don't have necessarily an ALS mouse. We have a SOD1 mutated mouse. And people use it as an ALS mouse. Obviously, it doesn't recapitulate all phenotypes, but I still think in ALS it's a pretty decent model to go for if we consider the other options.
All right, we have just 2 more officially scheduled minutes to ask Sophia for something with turned on translations stream. So if you have any question you didn't ask yet, that's your time.
Good evening. Can I ask another small question?
Please.
Go for it.
What about a sporadic form of ALS? Could it be a spontaneous de novo mutation arising in a new human, but in those specific genes? Or you or another researchers haven't detected those mutations in targeted genes at all?
So yes, of course, this is something. I think this is a matter of progress. So we will still, I'm sure, we will still find more genes that develop or that associate with ALS. But there will always be a part that will be sporadic, in my opinion. So I don't think that all of them are due to the, genetic mutations. And then it gets actually interesting. But yes, I think there are still many that we just don't see, that we don't know yet. Yes, for sure.
Interesting, thank you.
Okay, great. And now it's 1 hour and 30 minutes on the line. And I think with this, we can stop the translation and probably discuss a few more minutes if somebody has any question regarding professional advice or personal interest of Sophie or something like this. So thank you everyone for watching us. Can you say a few words for our followers on YouTube?
Oh yeah, well, I need to say thank you that everyone listened to my passion for ALS, and that you guys really have also in private time and in your spare time really take the chance to educate and to see more of what there is, because science is really big and there's still a lot to see. So I think the thank you goes to you guys that you have the discipline to watch that and be interested in that.
Thanks a lot. Subscribe to Sophia's blog and see you soon.